was like, we went too late. Too late. And she did an hour and a half sleep, so that's even 3 o'clock. I want to give myself ample time to get I don't know if they on them or you wait for them.
Okay, let's go ahead and get started. Okay. So just on the other class front, in PT cardiovascular, the hypertension lecture I did on Monday, it is posted now, so it's in the media site, not in the online lecture tab. So go to the media site tab, so it's in there. And then in that class, I also went through the cases, and I just this morning posted in the handout section the slides that had all the case answers to them. So when you go through the third part of the lecture first, then pull out the cases at the end so you don't have the answers when you go through them the first time, but you'll have them so you can review. Okay. So Suzanne, have you been in Alaska? I see you have Alaska the Last Frontier shirt. Are you an Alaskan person?
No, but I have been there.
Okay, so where have you been in Alaska?
I don't
That was quite a weekend. I was in Igigik, okay?
Where were you?
What do you mean, where? Igigik.
Oh, yeah.
It's right between Chiknik and Naknek.
Okay.
That Igigik. So there's Igigik. So it's not on the peninsula there. So if you kind of zoom in, there's Igigik. We keep going in. It's a little canning town. And the thing you'll notice is there are no roads into Igigik. So you can't get there or get away, quite honestly. You can only fly in on little tiny planes or come in on a boat, which really isn't much of an option. And so this is where I spent all of my summers when I was in pharmacy school, because this was the cannery right there. That's my cannery right there, okay? I was looking for a picture online that would best capture the nuances, the personality of Igigik. I think this one did it the best. That's pretty much what Igigik felt like for 4 summers of my life. So probably you didn't go to Igigik, right? Okay. All right. Well, I thought maybe we were a kindred soul there. And next time. Okay. So we're going to go through. So last week we went through EKGs. And this week we're going to talk about doing the physical assessment now of the heart. And then we'll combine both of those in the lab next week. So remember, next week we're going to be doing those EKG tracings that I handed out last time, that packet. The first part of lab is going to be I'm going to hand out some unknowns, and you're going to have to kind of figure out what they are. So please spend some time between now and then going through that packet, because it'll make the little tutorial that we're going to do at the beginning of that lab much more meaningful. If you come in just not having reviewed it, it's not going to help you a whole lot, okay? All right, so structure. So we're looking at the heart, sort of the structure of the heart. It's obviously that the center pump of the cardiovascular system weighs less than a pound. and pretty small, about the size of your closed fist during systole. So when it's contracted and the chambers are empty, about the size of your closed fist, and it has to work all the time, every time, 100,000 times a day. It's going to beat over the course of however many years you're living. So we really ask a lot of this little fist-sized shape, and there's no redundancies built in. It's one of those systems where there's no redundancies. Your liver, way over-engineered. You need maybe about a quarter of your liver, realistically, to do the majority of the functions you're going to need. Your pancreas, you can destroy 75% of your pancreas and still have relatively normal pancreatic function. Or we have redundancy built in. We have things like 2 kidneys instead of 1. Or we have 1 heart that can't regenerate, that can't do much healing of itself, and there's only 1 of them. And so that's why it's such a kind of a critical feature, and why we have so many cardiovascular-related diseases is it's the only thing we're dealing with. It has to do a lot of work. It's got to pump blood through about 60,000 miles of blood vessels. And it gets some help from your muscles and the return of that blood back to the heart through the venous system, but it's the only mechanism by which we're pushing oxygenated blood out to every cell of your body. So kind of going through a little bit of overview here of our structures, we have the right atrium. So we're going to be looking at the return of that blood through the inferior and superior vena cavas into the right atrium. It then kind of gets pushed over to the right ventricle. Ventricle then pushes it up to the lungs where we can get it oxygenated or exchange that gas, and it comes back into the left atrium and then into our left ventricle and then pumped out to the rest of the body. So we have our 4-chambered heart mode. The right side primarily responsible for returning blood to the heart and pumping it to the lungs, and the left side returning it from the lungs and pumping oxygenated blood out to the rest of the body. So the kind of the features here that we end up assessing most of the time is the function of these valves. When we're listening to heart sounds, we're listening to valvular sounds. Most of the times when we're hearing abnormal sounds, it's a result of abnormalities within the valvular system as well. So we have the tricuspid valve. The tricuspid valve is what separates the right atrium from the right ventricle. So you have that holding chamber where blood is coming into the right atrium, and then it's pushed into that right ventricle. The mitral valve is the one between the left atrium and the left ventricle. The pulmonic valve is between the right ventricle and then up to the lungs. And then the aortic valve is the valve that separates the left ventricle from the rest of the body. Where'd it go? Oops, there it is. There it is. So the valvular things are what we're going to be paying attention to. When we're listening next week in lab to hearts, we're going to be placing our stethoscopes over those areas where we can best hear those 4 valves in order to listen for abnormal valvular functions like murmurs. All right, so some other structures we're going to talk about. The apex and the base. What does base usually mean? When you think of a base of something, it means the bottom. Well, that's not the case here. The apex is actually, the apex is down here. So the bottom is called the apex. The pointy part down at the bottom, that's the apex. The base is actually the top of your heart. So it's kind of backwards from what we think. Base is on top, apex is on the bottom. We have the interventricular septum. That's this big muscular wall between the left and right ventricles. And it is the source of some other murmurs that can occur, because you can have holes between those ventricles. And so that interventricular septum can have a defect in it and allows blood to flow in a direction it's not supposed to go. The aorta is this coming out of the top here. Okay, so that's where the blood is going to be pumped out to the rest of the body. And we have these 3 big vessels here, brachiocephalic, left common carotid, and I can't remember what that one is. I want to ask you those, but these 3 vessels are kind of the first oxygenated blood that gets delivered. And where is it taking it to? It's taking it to your brain. So those, one of those or 2 of those vessels actually are responsible for getting oxygenated blood up to your brain. That's the priority that we put on that. Inferior and superior vena cavas are, so here's the superior vena cava. You can't really see the inferior one here. That are the vessels that return the venous blood from the body back into that right atrium. So if we're looking at sort of where the heart placement-wise is sitting in our chest, it's just underneath the sternum and then the left side. Okay, so left side is where the majority of the muscular portion of our heart is, right over the sternum and to the left of that on the patient's side. So let's look at the borders of this, trying to differentiate where the heart is. So the superior border of this, if you were to look at this, what landmarks that we talked about last week and the week before do you think would best describe that base of the heart? Rib 2. So okay. So what's the on the sternum at rib 2? Not the notch. The notch is up here. The angle. Good. So the sternal angle is where, and the second rib is about the level of the top border of your heart. Okay. So let's go down to the inferior border. Where would we describe that? So looking centrally here, where would this area be here? What's this? The xiphoid. So just above the xiphoid process, okay, that little bone that's poking down at the bottom of your sternum, just above that. Okay, so it basically covers, when you think about the heart, it basically covers the body of the sternum. Remember that sternal angle is where we have the manubrium connecting to the body of the sternum, and the xiphoid here is just sticking down just below the body of the sternum, so the full body of the sternum is about the size, top to bottom, of your heart. So the right border, how would we describe that right border? What anatomic line do you think that looks closest to? It's not really midclavicular, right? Because midclavicular is going to be over here a little bit more, okay? It's kind of just off the sternal border. There really is no perfect line for it, but if we had the sternal border being here, it's just off the sternal border a little bit. And so it just goes maybe an inch or so to the right of the sternal border. Then how about the left? If we were to describe this line right here, what line would that be? That's more midclavicular, okay? So that's where the heart sits. Now, this is basically how it sits in the chest there. So we have and this is a heart that if you ever go to see an autopsy or involved in seeing a surgery done that's an open heart surgery, this is not what you're going to see, because this is a really, really good looking heart, okay? The hearts you're typically going to see will look more like that, okay? Because we live in the United States and our diet means we have lots of fat. So we have a lot of fatty overlay on top of that heart. So this is how it sort of sits in the heart, in the cavity itself. But you can kind of see our portions here. This over here, what's that little thing up at the top here? That's the superior vena cava. So that's bringing blood in from the head. That makes this area right here, that's the right atrium. Here's our aorta coming out, the arch of the aorta with that brachiocephalic and left common carotid, and the one I can't remember, okay? It's coming off of there. And so structurally, it's sort of sitting in there, a little turned sideways compared to how we looked at it initially. Here it is in the cavity itself with the pericardial sac opened up and sort of folded back. And you can see, again, how much fat is overlaying that heart. That's a little more typical when you see a heart in somebody. Now, this is one of the things that you won't see very often, but we do have the capacity at some level to be able to replace some of the functions of the heart mechanically. And this is a really big dream about 20 years ago. And people worked on this really hard to figure out, this is just a pump. It's just a pump. There's nothing magical about it. It doesn't have a whole lot of things other than just pumping blood. Not a whole lot of hormonal control, not a lot of things that we need the heart to be able to do other than just pump. So let's stick a pump in there. And so in patients that had failing hearts, had failing myocardiums, who had heart failure, there was a big push to try and use mechanical hearts and to replace somebody's heart with a totally mechanical device. Functionally, it works fine, but there's just too many problems associated with blood clotting and rejection and the rest of it to really make it work. We also figured out that we can't design or haven't been able to design a pump that would last nearly as long as it would be necessary to have it replace somebody's heart. We just don't have the technical capacities to do what our heart already is capable of doing. So now we use them primarily just as a bridge. In patients who are waiting for heart transplants, we can use mechanical hearts as a bridge during that period of time, but that dream never really fully paid off. Let's talk functionally what's going on here. Now, most of the functions that we're going to be evaluating tend to be centered around the heart valves. And so here is, I think this is aortic valve. And so here we have a tri-leaf valve, where we can see we have one leaf here, leaflet, a second leaflet down here, and a third leaflet here. And that little line running between them, that's where they're sealed together. So this is a closed aortic valve. Ordinarily, which way is blood running through this valve? Is it running towards you or away from you? Looking at that valve, which way is blood going? So who thinks it's coming towards you right now? The majority of blood's coming towards you. Yeah, you're right. It's coming towards you. Okay, so the blood is rushing towards you, pushing those valves open, and this is sort of what we see. As we were standing over on this side of the valve looking this direction, these valves are relatively passive. They're not a muscular contraction causing opening and closing. They're a sealable flap. That's really what your heart valves are, is a one-way sealable flap valve. And so when the pressure on this side of the heart is greater, it pushes blood through the valve. And it just opens the valve through pressure. Now, once blood has rushed through to the other side and the pressure now on this side is greater than it is on the other side, blood starts to flow backwards because blood is going to flow where pressure takes it. As it starts to flow backwards, it catches these little valve things and pushes them shut to give us that closed valve. So it's like a parachute. As it's pushing back in, the parachute fills with air. This time it's filling with blood, and that forces those seams together. All these seams now, as it gets pushed back in, get forced together, and that's what seals the heart from retrograde flow. Now, this is another aortic valve, but this is an aortic valve that is a variant of normal. This is a bileaf valve. And so some people congenitally just have a bileaf rather than a trileaf valve, aortic valve. And this valve also has some pathology associated with it. This is in the closed position right now. So what do you see that's a problem with this closed valve? Yeah, it's got a hole in there, okay? So even when it's closed, blood is rushing the wrong way. That retrograde flow, that regurgitant flow, is going to result in potentially in symptoms, and we're going to be able to hear that through a murmur. Also, you get these big calcifications on the end. There's more stresses being put on this valve. So bileaf valves tend to not have the same physiologic life as a trileaflet valve, primarily because of the associated calcification that takes place. So here is that what we're talking about with calcification. As the valves, as they can fail, as calcification starts to form in, here you can see the ring of calcification around that valve when you're looking at it. And this is looking at it from the top. This is the beginning of each of those leaflets, and each one has calcification on it. The calcification keeps the valve from being as pliable. It can't open all the way or close all the way as easily because it just doesn't have the smoothness anymore. That calcification brings in some rigidity that keeps the valve from being able to open all the way or close all the way. If it can't open all the way, what do we call that? That's stenosis. So a stenotic valve is a valve that's incapable of opening all the way. So now we're having to push that blood through a smaller hole than we're supposed to. That limits the forward flow of blood. And then if it can't close all the way, that's the regurgitation. Now where we have a regurgitant valve that allows the retrograde flow of blood. So here's another kind of valve you can see, which is a mechanical valve. And patients who have valves that are failing from one of many reasons, including something like bacterial endocarditis. Here you can see on the screen, you can't see this well, but if you look at the monitors, it shows a lot better. You can see basically how these valve leaflets are destroyed. The bacteria on them are destroying the valves themselves, and so this really has no physiologic function anymore. The blood can pump both ways, and that can cause some pretty significant symptoms, including heart failure. So we got to get rid of that valve, and one of the things we can do is put in a mechanical valve. We can put in pig valves, and we can put in mechanical valves, and both types of valves are used. So the way this valve works is you can kind of see at the bottom down here, there's the hinge. And the way this valve works is just the same way. Instead of having three valves, it just has one. As the pressure on the other side builds up, it just opens up from that hinge. When pressure comes back, it catches it, pushes it back shut. Nice, easy mechanical valve. You can have other ones that are like gates. You'll have a top and a bottom, and as the blood, the blood will push in, it will like flap it open and flap it closed. So the same type of thing, just a mechanical way of being able to ensure the forward flow of blood without retrograde flow. Now, this stuff around the outside, this is why this valve had to come out. As you can see, this little tissue around the top part of the valve, and this was the tissue that was actually all the way around that valve. So what happened to this valve? Why did it have to get taken out? It couldn't open. It was a stenotic mechanical valve. Look at how this tissue overlays the valve itself. So you can see here's the border of the valve. The tissue comes all the way in. That valve has to open all the way towards you right now. It has to fold all the way back down to allow blood. it was running into this tissue, so it could just open up a little bit, some blood flow could come out. So this valve had to be removed and replaced because of this inflammatory tissue that starts to grow around the perimeter of the valve. It's just a bummer. It's an inflammatory tissue that can occur over a long term. It's the same type of tissue. When you look at it histologically, it looks a lot like the tissue that you can see in a patient who has rheumatoid arthritis. It's a tissue called pannus. It's a very inflammatory-based tissue, and that's what it is building here. So it's an inflammatory process that just ends up ruining the valve that's been replaced. So let's talk about what we're going to do to try and evaluate through an examination process the different functions that we've talked about. So we're going to be doing inspection. We're going to look at the height of the column of jugular venous blood. So we're going to look for jugular venous pressure increases. We're going to look for any abnormal chest wall motions that can tell us that there is substantial turbulent blood flow occurring below. We're going to palpate. We're going to find the carotid artery pulse. We're going to feel for something called the apical impulse, which is where the chest is basically beating, or the heart is beating against the chest wall. And finding the apical impulse can give us a bit of a sense as to how big their heart is. Not from a generosity standpoint, but an actual physical size of their heart. And then we're going to palpate along the base and the sternal borders of the heart, feeling for turbulent blood flow. And then the primary examination of the heart typically occurs through auscultation. We're going to be listening for abnormal sounds, listening for abnormal blood flow, which is a murmur, listening for abnormal heart sounds like S3 and S4 sounds, opening snaps, clicks, all sorts of little things that we can hear there. And primarily we're going to hear those over these valve areas. So our first thing is we're going to inspect. So ideally, we have to have some light source that's kind of coming from the side. We're trying to create shadows on the chest wall. And it's like if you shine a light directly on something, you can't develop any shadows. So when it moves, there's no contrast for your eye to catch. So that's why they always have those gooseneck kind of lamps that are in exam rooms, is to be able to throw tangential light against something where you're trying to look for motion or movement. And that's what we're trying to do here, is look for any abnormal movement. We're going to be looking for something called the apical impulse. So that apical impulse, is where the left ventricle is closest to the chest wall, and as your heart beats, it actually hits against the chest wall. And so we're going to look for that. Usually you're not going to see it. In some people you can, if they have a very hyperkinetic heart, or they have hypertrophy of their heart, or they have a really super thin chest wall, you can see it. But ordinarily you're not going to see the apical impulse. We're going to look for it. Now, where you want to look for it is where you expect to see it. Where you're going to expect to see it is in the fifth intercostal space in the left midclavicular line. So that's one of the places that we're going to be needing to get to, because that's where we're going to be looking for the apical impulse, feeling for the apical impulse. It's also where one of the valve sites is. So the fifth intercostal space, left midclavicular line, is an important place to get to. We're also going to be looking for things called heaves. Heaves are large chest wall motion. So it's an area that's really moving a lot. And we don't really have a specific cause of that. There are a lot of things that could cause heaves, but we're looking for these rhythmic, large areas of chest wall movement to tell us that there's something hyperkinetic or hyperdynamic happening in that heart. Then we're going to palpate, because as we get to murmurs, we're going to start grading these murmurs based upon how loud they are, and then whether we can feel the murmur, and lastly, whether we can see the murmur. So the highest grade murmur is one that there's so much turbulence occurring that when I look at the chest wall, I can see the chest wall move because of that much turbulent flow. The next grade down in murmur is I can hear it really loudly. I can't see it, but I can feel it. And so we're going to palpate for these murmurs as well. So we're going to try and palpate over very specific locations. We're going to palpate over the areas that we're going to best be able to feel turbulent blood flow happening at a valve. So one of the places is feeling for the apical impulse. So where did we say the apical impulse should be? Fifth intercostal space, left midclavicular line. So when they're laying on their back, we can try and feel it. Now the trouble is, when they're laying on their back, gravity is taking their heart away from where we're trying to feel. So sometimes you can't feel it very well because gravity's pulling their heart away from us. So that's where you'll see the examiner will oftentimes roll the patient onto their left side or have them sit up to try and bring the heart closer to the chest wall to see if they can feel that apical impulse. The location of the apical impulse can tell us about the size of the heart. In somebody who has cardiomegaly, who has a hypertrophied heart, what's going to happen is that PMI, the point of maximal impulse, or the apical impulse, will shift. It'll shift down and laterally. So now it's no longer in the fifth intercostal space, left midclavicular line. Now it's further out and further down. It's a kind of a down and dirty way of being able to feel or trying to assess the size of the heart. So here we have the feeling at the fifth intercostal space, left midclavicular line. So you can see it's basically right under the pectoral muscle, right under the breast. So in lab next week, we'll work on some techniques for how to get to this without being problematic, okay? So, in order to feel on a female, we'll talk about how we can do that in an effort to be able to get to the apical impulse, but not cause too much discomfort. So this is going to be where we're going to feel for an apical impulse. It's also where we're feeling for a particular valve, the mitral valve. The mitral valve is best heard and felt at that apex. So when you're at the fifth intercostal space, left midclavicular line, that's where we're going to be feeling for and later listening for the abnormalities in the mitral valve. So our next place is along the sternal border, okay, so the left sternal border. And again, we're feeling for those big movements or vibrations that tell us that we have a abnormal turbulence occurring. So a vibration is called a thrill, and those diffuse movements are called heaves. So now we're feeling along the left sternal border, because there are two valves along that left sternal border that we're going to talk about. And so there's one up at the second intercostal space and one down at either the fourth or the fifth intercostal space. We can feel the vibration that's occurring as a result of that valvular function along the left sternal border. And then lastly, we're going to feel at the base. Okay, and the base for the same thing. There are two valves we can feel along the base. On one side, at the second intercostal space, left sternal border, and right sternal border are two of the valves we're going to be feeling for, the aortic and pulmonic valves. So we're basically feeling over those areas where we are expecting the vibrations from abnormal blood flow through a valve to be greatest. That's really what we're trying to do here. Now, one thing we can measure is an estimate of right atrial pressure, and we do that by assessing somebody's jugular venous pressure. So the jugular vein is connecting to the right side of the heart, and so when we have blood backing up behind the right side of the heart, it backs up into the jugular vein. Do you have a question? Yeah.
Why the palm?
Trying to cover a bigger area. You could do it with two isolated places of fingertips as well. But if you use the flat portion of your hand, you can feel both sets of valves with one hand placement. That's probably the biggest reason. But if you can't do that or don't want to do that, you can place your fingertips over one and fingertips over the other. The palmar surface of your hand is good for feeling vibrations. Fingertips probably the best, but along your whole palmar, this is the palmar surface, not just the palm, but the palmar surface of your hand can feel vibrations better. So what we're going to do here is we're trying to figure out what the pressure inside that right atrium is by looking how far blood is backing up into the venous system. And the veins we're going to be able to measure that from are the internal jugular veins. So that's what we're trying to look for. Now, ordinarily, when we're in lab next week, nobody's going to find an internal jugular vein because I doubt any of you have significant enough heart failure that it's going to be sticking out. I hope not. If we do, that we'll have to call the hospital because that would be unusual. So we're going to have this vein that we're looking for, and when we find that it's engorged, that tells us that there's the potential for increased right heart pressure. Then we're going to determine whether it exists. So what happens is we're going to use the sternal angle. Remember, that's the place where we have the fusion of the manubrium to the sternum. That sternal angle is going to be what we're going to measure from. It's called our 0 point. Because regardless of what position we're in, and I'll show you a graphic here in a little bit, whether we're lying down or sitting up, in that position, that sternal angle stays a fixed 5 centimeters above the right atrium, or a fixed distance. In most people, it's about 5 centimeters, but it can vary depending on the size of the person. So it's a fixed distance between the right atrium and that sternal angle. So we're going to use the sternal angle. We're going to measure from the sternal angle. Now we're going to, basically, we need to put the patient in whatever position they're in that we can see the top of that column of blood. So let's kind of look at this picture for a second. Go over here so you can see it in the back a little better. So what we have is this middle line right here. As you can see, that's the sternal angle when they're at 30 degrees. It's over the sternal angle at 60 degrees and over the sternal angle at 90 degrees. So if we lined somebody up according to their sternal angle, that's kind of what this picture is doing. Now, when you go down, this is the center of your right atrium. So you can see the center of the right atrium when you're at 30 degrees stays a fixed distance above or below that sternal angle. It's the same distance when they're at 60 degrees. It's the same distance when they're at 90 degrees. So since we can't stick a ruler down into the right atrium, but we can stick it on top of the sternal angle, and we know the distance between the right atrium and the sternal angle stays the same, cool. We can actually measure how high something is above the right atrium by measuring how high it is above the sternal angle and adding 5 centimeters to it. Now you can see that this patient has, this is the internal jugular vein that we're looking for, okay? So when they're laying down, and when this person is laying down, and this is going to vary depending on how high the pressure is, but in this particular example, this patient is laying down at that 30 degrees. I want to try and find the top of that column of blood. I'm trying to measure how far the blood is backed up above that right atrium. So I need to find the top of that column of blood. It's like blood filling up, it backs up into a column. So I really can't see where the top of that column of blood is because it kind of disappears behind his jaw here. So that position wouldn't really work for me. So, okay, so I'm going to have him sit all the way up. So the same patient in the same circumstance sits all the way up. Well, now gravity is pulling that blood down here. And so in this circumstance, it's too far down. It's kind of behind the clavicle. So I can't get to it there either. So I lean the patient back until I can see the top of that column of blood. And once I have the top of the column of blood, I measure how high it is above the sternal angle, and that's my jugular venous pressure. So I'm trying to estimate how high above the sternal angle that column of blood goes. Now in lab next week, since you won't have that column of blood, you won't be able to measure it. So we'll pretend. Okay, we'll draw a mark on your neck and say that's where the top of the column of blood is. Now measure it. So here's how we're going to do that. We can't measure, so if the column of blood is over here on his neck and the sternum's over here, how do I measure how high this is above that point? I have to use 2 straight edges essentially, a ruler and a straight edge. So I'm placing this ruler on the sternal angle because I'm trying to measure again how high that column of blood is above the sternal angle. So this ruler goes in the sternal angle and it has to be absolutely perpendicular to the floor, straight up and down, because I'm measuring a height, okay? Now over here, let's say this is the top of that column of blood. I use this to connect that top of that column of blood to the ruler to measure on the ruler how high it is above the sternal angle. So if this was higher up in this person's neck, it would be higher up on the ruler. So this ruler has to be absolutely parallel to the floor, and they have to meet at a right angle. Now I'll show you this in lab. It makes a lot more sense when you kind of see it in front of you. But it's essentially, how would I measure how tall, I'm going to make up an example that's bad, so I just won't make it up. Okay, so here's a picture that has a little line here. This is that internal jugular vein. And so you can see that it's backed up, it's backed up, it's backed up, it's backed up. This is the other portion of that vein coming up. And so you can see the vein as it's coming up right here. This is kind of where I can't see it anymore. It's right before it goes behind the jaw. So if I wanted to get this to the point where I could see that better, what would I need to do to the patient's position? Have them sit up more to allow gravity to pull it back down more. So you can see how engorged this vein is with blood. You're also going to see it doing a pulsation. You're going to watch the pulsation. You can't see it on here because it kind of shows up and disappears and shows up and disappears as you see that pulsation. I'm trying to find the top of it and measure that above the sternal angle over here. see, did I put on here? Okay. All right, so our next mode here is where so far we're doing things that we're only going to be probably doing if we've heard abnormalities on cardiac auscultation, okay? Because that's going to be our more sensitive measure of the presence of a murmur or of an abnormal valvular sound or activity. So cardiac auscultation. We want to listen over the valvular areas. So that's one of the things you got to get good at next week in lab is being able to estimate where the valvular areas are so you can place your stethoscope over them and have the best chance of hearing any abnormal functions. We're going to be using both the bell and the diaphragm at each of those valve sites because we're going to see some turbulent flow is going to be those murmurs are more likely to be lower in pitch where I need to be able to use the bell of my stethoscope, but others can be higher, like an abnormal heart sound, like a third and a fourth heart sound can be a little higher pitch, so I want to use the diaphragm to find those. So you need to make sure you're listening at both areas. We're going to be assessing the normal sounds, and if there are presence of any abnormal sounds, and listening for rate and rhythm as well. So the sounds we're going to be listening for that you're going to hear in everybody are going to be these sort of 4 sounds or periods of listening. We're going to have they name these things very, very unique. The first heart sound is called S1, and the second heart sound is called S2. Guess what the third heart sound when it's present is called? S3. Very imaginative. So S1 is going to be, everybody's going to have that. Okay, so you're going to hear that. Everybody should have S2, so you're listening for those 2 sounds to hear whether they are normal or not. We'll talk about some abnormal variants. Then you're listening during the period of systole and listening during the period of diastole. So let's kind of go through those sites first, and then when and how you're going to listen to those things. So our valvular sounds, where are the auscultatory sites you need to listen to? You're listening over the 4 valve areas like we talked about when we were feeling for them during palpation. So our first valve that we're going to talk about is the mitral valve. And so the mitral valve is at the apex, also where the point of maximal impulse is, or the apical impulse, all those terms roll together. The 5th intercostal space, left midclavicular line. So this is one where you're going to take your stethoscope and kind of push it up underneath the pectoral muscle or up underneath the breast in order to get to that 5th intercostal space. The next one is the tricuspid valve. The tricuspid valve is along the left sternal border at the 4th intercostal space. This is why you needed to get good at finding the sternal angle, finding the 2nd intercostal space, because the 2nd and 4th, you're going to have 3 valves that are present at those areas, okay, so that you can get to easily on everybody. The pulmonic valve is at the 2nd intercostal space on the left sternal border. So we find our sternal angle. We come just off of that. We find the 2nd rib there. Right below that is the 2nd intercostal space along the sternal border. That's you're going to hear it. And then our last valve is the aortic valve, and it's just across the sternum. It's at the 2nd intercostal space on the right sternal border. So how we're going to listen to these is usually in along the line that's formed there, okay? So probably the easiest place to do this is to start at the aortic valve. Find that sternal angle, go just to the left of it, down into the intercostal space, and you can find in that 2nd intercostal space, that's where you place your stethoscope. Make sure that you're off of the sternum. You don't want to overlap more than maybe a quarter of your stethoscope with the sternum. So a big mistake people will make is they overlap the sternum too much, and you're not going to hear much through bone. So you've got to move it off the sternum enough to hear it, but not so much that you get clear over here where you don't need to be. So we're just off of the sternal border is aortic. So that's an easy one to know where you are. So you find that sternal angle, you find the 2nd intercostal space, you place it there and listen. When you're done listening there, you just jump across the sternum into the 2nd intercostal space on the left sternal border. So now you're listening to the pulmonic valve. So again, you can get exact on those, and all you have to do to be exact is find the sternal angle. So when people were having problems last week, they're finding the wrong sternal angle. This is where it's going to be a problem. If you think that that first bump you felt on the sternum was the sternal angle, you're going to be up here. and that's too high to really hear the pulmonic valve. So we're going to go through that again, make sure you're accurate. In lab next week, you should try and find more than just your lab partner's sternal angle if you can, because again, they won't be there on the exam, and you're going to need to make sure you can accurately get to sternal borders. So now we're going to be listening down along the sternal border, because even though that tricuspid valve is down here at the 4th intercostal space, it might be 4th or 5th. And so we're going to listen down along that sternal border until we get to the tricuspid valve. So we're listening along the border, one stethoscope width at a time, because you're going to have alternate sites for the tricuspid valve that may not show up exactly where you think it is. And then once we get to the tricuspid valve, then we can basically listen along the border until we get to the mitral valve. So we're listening along the left sternal border line and wrapping around to get to the mitral valve, because there is anatomic variability, especially in people who have cardiovascular abnormalities. If their heart is shaped, if it's bigger, they have cardiomegaly, then we're going to hear those sounds at slightly different places. So we listen along that entire line. So that's kind of the sequence. You can start on either end, but I think it's easiest to start up here because you have a bony landmark right there that's going to help you find those two valves where you can be absolutely certain. There's enough variability for, if you just go straight down to right underneath the pectoral muscle or right underneath the breast and say, I know that's 5th, it's probably 5th, but you don't know that it's 5th. It could be 6th. So that's why starting at the top and working your way down tends to be more accurate in being able to find those sites. So we have where we're going to listen. Now we need to talk about what we're listening for. Okay, so what are the sounds we're expecting to hear, and what are some of the abnormal sounds that you might hear? So our first sound is the first sound, S1. And this sound results from the closure of the mitral valve and the tricuspid valves. Okay, so mitral and tricuspid valves. And where was the mitral valve? 5th intercostal space, left midclavicular line. Where was the tricuspid valve? 4th intercostal space, left sternal border. So they're both down here. So where do you think that sound is going to be loudest? Down here. Now, let's kind of go back for a second. So if I took in fact, let's go to this one. Okay. So we talked about where anatomically how the heart was sitting in the chest. What was the superior border? Sternal border, okay? So the sternal angle, okay? So you have the sternal angle there is where the top border is. And what was the right border of the heart? Just off of the sternum. So do you think if I took a drill and I drilled straight down there, I'd hit the aortic valve? Where was the aortic valve actually located? It's actually located over here. That's where the valve is. Is that where the mitral valve is? That's the apex of the heart. That's the very pointy part of the heart. So the mitral valve is probably over here. So why am I listening at those areas? If that's not where the valve is, and I'm trying to hear the valve, why am I listening at areas where the valve isn't? It's like when you were a little kid, okay, and you had those long, it's Christmas time or birthday time, because those long tubes that the wrapping paper's on, and so you just play, you whisper in one end and hear it out the other. That's exactly what's going on. If I'm whispering into the end of that tube, where are you best going to hear my voice? On the other end of the tube. This is the other end of the tube. Physiologically, this is where these sounds come to the surface. It's not where the valve is located, per se. It's where the vibrations associated with that valvular function reach the surface. That's why we feel there for the vibration when we're doing our palpation. It's also why we put our stethoscope there to listen for them. It's not physically where the valve is located. It's where the sounds of the valve are going to make it to the surface. That's what each of those 4 valve areas are. So let's go back. And S1 results from the closure of the mitral and tricuspid valves. So we're going to hear it louder where those valves sounds reach the surface. So it's going to be louder at the apex. Remember, the apex is the pointy part down at the bottom. The base is up at the top. So aortic and pulmonic are at the top. Mitral and tricuspid are down on the apex. It's going to be a higher pitched sound. Both S1 and S2 are higher pitched sounds, so we hear them best with the diaphragm. And if you're having trouble, because some people are going to have lots of murmurs and lots of other sounds, trying to figure out what's what can be confusing. So you need a starting point. A starting point can be S1 because it's always going to coincide with their carotid pulse. So if there's a whole bunch of sounds going on and you're just not sure which one's what, feel their carotid artery, listen over there, and the sound that's occurring in conjunction with that pulsation, that tells you that's S1, and that's your starting point. So patients that have loud S1s, well, if their valve is stenotic, so if that mitral valve is stenotic, which is much more commonly than a tricuspid valve being stenotic, because it's on the left side of the heart, it's got more pressure going on. So you can hear it there, and you can hear it primarily in patients that have a really high heart rate. You can hear a little bit louder S1. And that's the same thing with these hyperkinetic states, basically hyperthyroidism, those types of things that have a really fast metabolic and heart rate going. So we're going to look at these things sort of on this graph where we have the sound, and the loudness of the sound it coincides with the height of this bar. So the first heart sound is heard louder at the apex than it is at the base. Second heart sound. Second heart sound is resulting from the closure of the other two valves, the aortic and the pulmonic valves. So those are second intercostal space, left and right upper sternal borders. So you're going to hear them louder at the second intercostal space, right and left upper sternal borders. So we're going to hear it louder at the base than we are at the apex. And typically a louder than expected S2, the most common occurrence is going to be just in patients who have hypertension. Not just high blood pressure acutely, but hypertension, chronic hypertension. So now we have our S1, okay? Let's hear, that's our second heart sound, so let me click it here. There we go. So we have S1, and then following that is S2. At the apex, it's going to be not as loud because that's not where the valve is. At the base, it's going to be louder. So let's listen to the apex first. You're going to hear S1, then S2. And what you should hear is S1 louder, S2 less loud. Now, let me make sure our volumes are nice and good here. Okay, let's see if you can hear this or not. We may have to crank it a little bit. Come on. There we go. Getting a little extra there. A little distortion. Okay, I'll turn it down a little bit here. Okay. So you heard bump, 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 bump. First one's louder because we're listening there to S1 at the apex. You're going to hear S1 louder than you are S2, just physically where it's located. Now, they're not labeled, right? There's two sounds. How do you know which one was S1 and which one was S2? The timing is going to be the thing that's going to guide you a lot. So let's think about what takes longer to occur. Filling of the heart or contraction of the heart? What's longer? Filling is, okay? So S1 is the sound that marks the beginning of systole. And S2 basically marks the end of systole or the beginning of when filling is going to occur. So when you're listening, you have bump, bump, and then a longer pause, bump, 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 bump. Your ear naturally hears that first sound as the sound that occurs after the long pause. 1, 2, 1, 2, 1, 2, 1, 2. So your ear clues in that first sound after the pause, that's S1. The sound that follows right after that, after a short break, is S2. Then there's a longer pause and it starts over again. So listen to it and you can intuitively just know which one's S1 because it occurs first after that long pause. 1, 2, 1, 2. Okay. So let's go to the base. Same two sounds, only now we're listening to them at the base. And what valves are at the base? The valves associated with our second heart sound. So we're going to hear it louder there. So the first time it was bump, 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 bump. This time it's bump, 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 bump. The loudness of it can kind of tell you, or you should expect to hear, S1 louder at the apex, S2 louder at the base. Okay, so we're done. S1, S2. Well, unfortunately, we're not done because S2 has some interesting things associated with it. S2 can actually split. Okay, so S2 splitting is the circumstance where remember S2 is from the aortic and the pulmonic valves, okay, the ones up at the top here. It's from them closing. And what if they close at slightly different times? We can hear that two sounds instead of one, and that's what splitting means. We're splitting the one sound that's caused by these two valves closing into each of the valves individually, so now we're going to hear two different sounds. Physiologically, this is normal, and you're going to hear it when somebody is during inspiration and until they exhale. So they breathe in, that's when you're going to hear the splitting more. So we're going to listen for some splitting next week. It's hard to hear in the lab just because there's always some noise, but take your stethoscope out this week, okay? And just when it's quiet, just listen to your own heart and listen to it and see if you can't hear while you're lying down, relaxed, comfortable. See if you can hear that splitting S2. It's going to be heard best at the right upper sternal border and the left upper sternal border. That's our second intercostal space, right and left sternal borders. Okay, so we have during inspiration, oops, I'm getting my pals here. So during expiration, the sound, basically S2 sound, just sounds as one sound. After inspiration, it splits into these two different sounds. So let's listen to S2 splitting. Expiration. Inspiration. Back to expiration. Did you hear it? Yeah, you did. You just don't know it yet, okay? So think of it as this. Think of it as, that's two sounds, right? But they're happening at the same time, so it kind of sounds like one. But you can tell that's one and that's two. That little difference is what you're going to hear in splitting. What you just heard on this thing is like a pretty wide split where it goes bump, ba bump, bump, ba bump, bump, ba bump. So listen to it again. You're going to hear it bump, 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 ba bump, bump, ba bump. Hear it? Yeah. Bump, 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 rather than just bump, 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 bump. And I hate to tell you, but that's a pretty wide split right there. Nope. Now you can have a wider split. That's what you just heard. That's a little bit more than physiologic. I really should put that under wide splitting and not physiologic because that's a pretty big one. Think of it as a split, as the sound, instead of it being sharp, as being kind of muddy. And instead of, that's a sharp sound. That you can hear is not quite as sharp. It's thicker, it's muddier, there are terms to describe it, but it's not that level of sharpness that you expect to hear when they're occurring simultaneously. We also can have fixed splits. So a fixed split is it doesn't matter when they're breathing, you can still hear the split during inspiration and expiration. Hear the sound? Bum 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 bum. So they're breathing in, they're breathing out, doesn't matter, you can still hear the split on both sides of it. That's a fixed split. And then lastly, one that you'll never hear, it's really rare, but you'll hear about a paradoxical split. A split occurs at the wrong spot. So it's just the opposite. Instead of occurring after inspiration, it occurs after expiration. So it's just a rare beast. Okay, So we have S1, S2, and some variants of S2. Yeah.
With the wide and the fixed paradoxical, are those pathological or are they just abnormal?
They are typically pathological. I'm wondering, I'm trying to think if there are circumstances where they can just be abnormal, not pathological, and I'm not sure. So now we have our extra heart sounds, ones we're less likely to hear in lab next week. S3. So an S3 is a lower pitch sound. Okay, so we're still going to hear it with both the diaphragm and the bell, but you'll hear it in comparison to S1 and S2, it's a lower pitch. And so it's heard best with the bell, because it's that lower pitch, and it's heard best at the apex. So where was the apex? Bottom. Okay, so it's down here at the fifth intercostal space, left midclavicular line. That's our apex, where the apical impulse is. It's normal to hear an S3 in kids and in younger adults. It becomes abnormal kind of once we reach about 40. And the number one thing it's associated with is heart failure. So when you see in a chart S3, your mind should flick straight to heart failure patient, okay? Less likely mitral regurgitation, so don't even worry about that. Worry about you have S3 in the chart, your number one thought is heart failure patient. That's what I mean by the terminology. People won't say this is a patient with heart failure. They'll say this is a patient who has bilateral pitting edema, who has an S3 and increased jugular venous pressure. Everybody in the room is thinking heart failure. You got to be thinking heart failure too if you're going to be useful with regards to making recommendations for drugs. Okay, so here we have our S1, pause, S2, and then right after S2 is when we're going to hear S3. So let's listen to it. Bump, ba bump, bump, ba bump, bump, ba bump. Okay. You hear why they call these gallops? Okay, it kind of has that galloping kind of sound to it. Bump, ba bump, bump, ba bump. So what's the difference between a third heart sound and a split S2? How could I tell the difference? A split S2 is kind of in that same area, right? What's the difference? Well, if you have a widely split S2, they don't happen at the same time either. So the timing of them can be exactly the same. When you hear the sounds can be exactly the same. What's that? Well, it sounds different. Yeah. Why does it sound different?
Is it like the sharpness of it? Like they have the same, as opposed to being hollow?
A widely split S2 is going to still be very sharp sounding because it's separated from the first part of the split to the second part of the split. So what's the difference in this? Bump ba bump 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 bump ba bump. Could you hear the difference? They sound different? Bump ba bump bump ba bump bump ba bump bump ba bump. It's the pitch. What's going to be bump ba bump bump ba bump bump ba bump? Which one's that? Split S2. That split S2, both of the sounds are the same. The valves closing are going to sound the same. They're just split apart. Bump, ba bump, bump, ba bump. But the S3 is lower in pitch. Bump, ba bump. So it drops down. The timing's the same. Bump, ba bump, 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 bump, ba bump. That dropping of the pitch. So that's one difference. How else could you tell the difference? Whether it's changing with the expiration or not. So regularly split S2 is only going to show up during expiration. It's going to disappear during inspiration, or the other way around. So we can hear it with respiratory pattern variation. So that's a way to tell the difference. Where are you best going to hear a split S2? Which valves is it from? Upper or lower? Upper. Okay, so you're going to hear it best up here. Where do you hear a third heart sound best? At the apex. So you can start to have differences, even though the sounds are rhythmically the same. Where you hear it, when you hear it, and the pitch of that third sound is going to help differentiate one from the other. Okay. We've got 10 of these, so settle in. Fourth heart sound, no, we only have 4, so just relax. So we are pretty close to being done. My vote is just to pile through for another few minutes and be done rather than take a break and come back for a few minutes. You okay with that? Okay, let's do that then. So the fourth heart sound is one that occurs clear at the very end. What was the very first sound in our cycle? S1. That's kind of what anchors us is S1. Then we have S2 and maybe an S3. S4 occurs all the way at the end of that. So if you think about it as a circle, it's occurring right before the circle starts again, which is at S1. So it happens right before S1. It is lower pitched, so you're going to hear it better with the bell than the diaphragm. You're going to hear it at the apex, just like we best heard our third heart sound at the apex. You're going to hear an S4 best at the apex as well. It's normal to hear in elderly patients. It's normal to hear in people that are very cardiovascularly fit, so athletes that have really good cardiorespiratory systems. When in other than those patients, the number one sort of thing that you think about, and you're going to hear it, is systemic hypertension. Patients that have significant systemic hypertension will frequently have a fourth heart sound. Okay, so we have S1, S2, and that S4 is going to occur right before S1. So let's listen to it. A lot of reverb. Ba-dum bum, ba-dum bum, ba-dum bum, ba-dum bum. When you're trying to figure these out early on, think of the rhythm of them. And so you'll see people when you're in the hospital and you're watching people, residents, medical students, listening to patient starts, oftentimes they'll be tapping, okay, to try and get a rhythm of the sound. Bum, ba-bum, bum, ba-bum, bum, ba-bum. They start to feel the rhythm of it, or ba-bum, bum, ba-bum, bum, ba-bum, bum. The rhythm can tell you a lot about which one it is. So, which one's this one? Bum ba bum 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 bum ba bum. What is it? Split S2. Okay. How about ba bum bum 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 ba bum bum. Hold up the number of fingers whether it's a 3 or a 4. Ba bum bum ba bum bum ba bum bum. Okay. How about bum ba bum 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 ba bum bum ba bum bum ba bum bum ba bum bum. It's the rhythm of it. So start listening for the rhythm of it. Once you have the rhythm of it, then you can figure out things like pitch and the rest of it. But if you can spot the rhythm first, that's going to help you to kind of get into that, where it is. Tapping, whatever it is, can help you to get to that point. Okay. Some people have all 4. I know it. Ba-dum, 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 ba-dum. Ba-dum, 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 ba-dum. 2 sounds, pause, 2 sounds, long pause, 2 sounds, pause. So they have an S4, S1, then the short pause, then the S2, S3, then the longer pause, then S4, S1 again. So this is a quadruple rhythm or a full gallop, right? Sounds like a horse. It's 4 feet, brum, 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 that kind of thing. That's why they call it an S or a quadruple rhythm or a quadruple gallop. Okay, so those are the primary extra sounds. We're not going to talk about snaps and clicks. Yeah.
Where do you see the full gallop, and is it sustainable?
It's basically, these are just valves that are closing at slightly the wrong times, but not necessarily enough to affect a lot of physiological function. But just like we said, if somebody had heart failure because of long-standing hypertension, well, there's your S3 from the heart failure and your S4 from the hypertension. And so that's usually what it is. It's kind of the cause of their heart failure has been sustained elevated hypertension. Yep. So that's one group of sounds. There's the extra sounds. Then we have murmurs. So murmurs, we've already listened for some murmurs. We call them murmurs when they're associated with the heart. What do we call them when they're associated with something else? A bruit. So a bruit is turbulent flow. Okay, so we're listening for turbulent flow associated with the heart. That's what a murmur is. So we have the different ways that we can generate turbulent flow. One is you can have a stenotic valve. That valve doesn't open all the way. And so the blood kind of rushes through the opening in the valve. There it goes. Rushes through the opening of the valve. And then once it gets past that opening, it expands into this chamber. And that's what causes the turbulence. So you can hear this when a flow across a partial obstruction. Another way is if you have normal valves. So those valves are supposed to be all the way open. So I couldn't really draw that very well. But they're all the way open, like normal, but there's just so much blood flowing through them, that causes some turbulence. So increased flow through normal valves. This is what you're typically going to hear in patients that are very anemic. They have such low red blood cell count, they've got to move that volume of blood around a lot more because they've got fewer red blood cells to move the oxygen around. And so that increased flow can cause murmurs. An aneurysm. An aneurysm is where you have a weakened portion of a blood vessel. Okay, so here a blood vessel that is ballooned out because the walls of it are weaker. So you have the blood going through and then expanding into that chamber. So that can cause a murmur. And then backward flow. So now that valve, the blood's supposed to be going that way, but when the pressure builds up on the right-hand side over here, it becomes such that it flows back through the hole and into this empty chamber back here and expanding into that empty chamber. That's a murmur from regurgitation. So blood flowing in an opposite direction that it should can do that. And then our kind of last one is where you have something called a VSD, a ventricular septal defect, or a valvular abnormality at birth called patent ductus arteriosus. So basically this is shunting. Blood's supposed to be going along this top one, but there's a communication with someplace else where it's not supposed to be there, this hole. And so now the blood, instead of just going where it's supposed to go, can go across this hole, and that causes turbulence. So remember that the ventricular septum, the thing that's between the right and left ventricles, if you have a hole in that, it's called a VSD. And a VSD allows blood to flow from one ventricle to the other ventricle and back, which is not supposed to happen. So you can hear that murmur, and blood is always flowing to the heart. So a VSD is a continuous murmur. It's just always going on. You're hearing it just flowing one way or flowing the other, but the turbulence is continuous, and so you can hear it that way. A patent ductus arteriosus, that's what you're going to see when you're on peds. And it's when we're born, we have a hole in the septum because we have to allow mother's oxygenated blood to get to baby. And then once the baby's born, that hole closes off on its own. Patent means remains open. So you have a ductus arteriosus that closes upon birth or within minutes after birth. But if it stays open, it stays patent. That hole stays there, and now blood's traveling in a way it's not supposed to. And babies can be born with heart failure. They're born, everything's fine. Within hours, they start developing heart failure because the blood is not moving through the body. It's just moving around in the heart. And so they'll go in and they'll seal it. They can do it with medications like indomethacin, or they can do it surgically. Nowadays, they can see it before it actually happens. They can do in utero surgeries as well. But that patent ductus arteriosus, or a PDA, that's what we're talking about with murmurs. Okay, so we're going to have to characterize these. You're going to hear descriptions of murmurs. So just knowing the terminology of murmurs. One thing is the timing of it, when it's occurring. So we talk the period between S1 and S2 is what period? That's systolic. And between S2 and the next S1 is diastolic. So if you hear a murmur that happens between S1 and S2, we call that a systolic murmur. If it happens between S2 and the next S1, it's a diastolic murmur. It's when it's occurring, either during contraction or during filling of the heart. That can tell you what valve is messed up or what type of defect it is. So that's an important distinction. Now, if your ears get really good, you can tell when it's occurring inside those periods. So if it happens at the very beginning of systole, we call that an early systolic murmur. If it happens at the end, we call it a late systolic murmur. And there's actually a third category of mid. So now you can have early, mid, and late systolic murmurs, or early, mid, and late diastolic murmurs. When you're listening to the sounds that I post on the Canvas site, you should be able to recognize a systolic murmur from a diastolic, but not beyond that. I don't care if you know it's early, middle, or late. But you should be able to hear, does the murmur occur between S1 and S2? Does it occur between S2 and S1? Holosystolic means it's throughout the entire systolic period. Holodiastolic means it's throughout the entire diastolic period. And continuous means it's happening all the time, regardless of systole or diastole. So that patent ductus arteriosus or that ventriculoseptal defect, those are continuous murmurs. They're just happening all the time because blood's always moving and you're always hearing the murmur. So one way we characterize them is based on timing. The second way we characterize them is based on pitch. Are they a high, medium, or a low pitch? The last way we kind of characterize them is on the pattern of the sound, okay? So we'll hear something called a crescendo murmur or a decrescendo murmur or a diamond murmur, okay? So let's listen to a crescendo murmur. Starts quiet, builds in intensity, like a crescendo. Or a decrescendo, starts loud and fades away. Or both of those together. It starts quiet, it builds in intensity, and then it goes away. Like an ocean wave. So it's characterized by the pattern of the murmur. And you'll see people actually drawing these types of things out in the medical record to signify the type of murmur and the timing of when they're hearing it. Nope. Stop it. Next slide. So other things, where you heard it best. Which location was it the loudest? Could you hear it in one place and it radiates to another place? So the movement of it. Whether it changes, whether they're breathing in or breathing out, or something called the quality of the murmur. Quality of the murmur are things like machine-like, or blowing, or raspy, or they're just words to try and describe the sounds that they're hearing and correlating it to something that we all kind of understand. Machine-like or mechanical has a certain type of sound to it. I would never expect you to be able to listen and differentiate these things, but recognizing that's the terms you're going to see being used in a medical record to describe different types of murmurs. And if you spend a lot of time in cardiology, those descriptions will start with them carry certain meanings as to what the cause was, but we don't need to worry about that. So here we have a few murmurs, okay? Common heart murmurs are valves that have gone bad. Those are the most common murmurs. So a mitral stenosis, mitral regurgitation, or aortic stenosis and aortic regurgitation. Why are mitral and aortic more common than tricuspid and pulmonic? These are left-sided heart valves. Left-sided heart valves have a lot more pressure put on them because that's the side of the heart that's having to push blood out to the entire body. Much more muscular, much more pressure being generated, and so valves in those areas tend to be easier to hear and fail more frequently. And so that's why we tend to hear those valve murmurs that are a higher pressure area. And the last type are those continuous type of murmurs where we have that it occurring the whole time. So here is a continuous murmur. In the background, there's all the time. You can't even hear S1 and S2 very well. It's blocked out by the murmur. So here is a holosystolic. You're going to hear a murmur, and then a pause, and then a murmur, and a pause. It's only going to be happening between S1 and S2. That blowing kind of sound. So when you listen, that's where the murmur is occurring. So as you're listening, just kind of watch up here. You can That's the murmur you're hearing right there. And then there's a pause where you don't hear anything. The murmur is happening between S1 and S2. Now listen really close this time and see if at the beginning of that you hear a bump and a bump. Listen for S1 and S2 and see if you can hear them in there. At the very beginning and at the very end, there's a bump, 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 bump. And in between that, whoosh, is that murmur sound. So listening, being able to hear S1 and S2 is going to be critical in being able to time those murmurs out. Okay. So on our website, when you go in, you can go to the audiovisual resources, and in there will be these sounds that we heard in here today. So we'll have a S1 and S2 at the apex and the base, a physiologic and persistent split S2. You hear S3, S4, and 4 different types of murmurs. So start training your ears. Now, there will be, on the second OSCE, murmurs and respiratory sounds that you will listen to and identify. I have a little speaker that you'll use your stethoscope and place it over it just like you'd listen to it on somebody's body. So when you're practicing listening to these things, practice listening at some point with your stethoscope over your computer speaker. It sounds different than your earbuds or than just having it open in the air. So listen with your stethoscope to get used to hearing the sounds that way, because that's how they'll be on the exam.
In general, with bruies, we were told that the bell is more effective. With murmurs, is that also true? And also for the speakers, does it matter which side we use?
Try them both. You're going to be able to hear different sounds through them, so definitely do both of them. You should hear lower pitch sounds better, whether you're listening at a speaker or listening on a body with the bell. So try it both ways. I will use sounds on an exam that are not subtle, okay? I'm going to use sounds that I think are big enough that if you spend some time with these, it shouldn't be a problem. But if you don't, it'll flux them. You have no idea what's going on. You just hear a bunch of sounds. You've got to spend some time learning the rhythms. Once you know the rhythms of it, it'll be pretty easy. But that's the prerequisite, is you have to be able to pick those rhythms apart. And I'll mix respiratory sounds in with cardiovascular, with heart sounds. So be able to do wheezes and rhonchi and crackles, and be able to differentiate S1, S2, S3, S4, and either a diastolic or systolic murmur. You do that, you'll do fine on the OSCE, but that will definitely be a part of the OSCE. Okay, that is it. We are done. So next week in lab, you've seen where we're listening to things along here, and so the dress is sort of like we did last time for respiratory. As long as we can get along the sternum and people can get to those valve sites, that's probably the biggest thing. All right? We are done. I'll see you in lab. Where are you? Oh, there you are. Gotcha. Thank you. Gotcha. Hey. You made it. Just inside.
Y'all are funny. We're not wrong.
Also, this one is really fun. You can delete it yourself. Flutter on your phone.
You know when they're like sitting there and you're having voices in your head and all of a sudden
So, but they like cut out all the vowels and they but it's and I changed it to
no, like it would have been like crawling. I gotta get out of here. That's fine. Don't worry about it. I can't. Literally, I was like, this is Kelly's fault. Because I saw him that one. You did. We were sitting for the Oski. Kelly summoned him, and I was like, what are you doing?
Call Suzanne something.
No.
Mental gymnastics. You want that support? It's like sitting on the doing it on it just bothered me because like it was always sitting on that.
Veronica, your computer's touching the thing.
Remember when we were banned from using that like 10 inch?
After long, we didn't know to do
No,
that.
they just
I don't even, okay, I don't know.
I don't
That's very cool. If somebody tries to get me, but everyone like, I don't want to touch it.
Oh, I didn't know that was a thing.
I brushed them very well for
Okay, I didn't
30
know that.
minutes. I know. Yes. Near Pittsburgh.
it's so fun. So it's just want to share with them. But I feel like nobody's gonna appreciate it. Yeah, see, it makes so much sense. Which Jessica is this? Is Jessica? Well, Jay Bird's not here. Nowhere does it say you have to be present. I don't think so. I'll have to double check that. Autopay.
You didn't bring your coat?
It's funny. You don't get it when I'm.
I'm almost like yeah, got one more.
We got it? Yes.
What?
That's me right now. I'm wearing a I've heard about it. I don't think that's I don't think you.
I was looking at it. So, you're on the other
The only thing on the template that doesn't have to with the study give you the only thing that doesn't have to do that is number 2. And number 2 is topic. So now you're the study is right here. That's good. And then number 2 so number 2 is the one you pick, topic is, and then one gets to have an objective. I was looking for it.
I had to put the objective as this. So, did y'all not have patient assessment this morning?
It's like you're really fun when you take it.